to Science Unplugged, the podcast where science and conversation collide. Join us for a deeper look into the world of innovation. We are your hosts, Charvi Trivedi.
And Taslima Farooq. Let's explore the insights from the world of science. From the lab to the field, we'll dive into the stories and experiences of researchers, experts, and innovators who are shaping our understanding of the world. Today we have with us Dr. Babukrishna Maniyadath. She's currently a postdoctoral fellow at Siddhant University, working in the field of molecular physiology. Welcome, ma'am. It is an honor to have you here today.
Hi, Charlie. Hi, Taslima. Thank you so much. Thank you so much for the welcome, and thank you for considering me for this podcast.
Ma'am, your academic profile is very interesting, and students who are on the verge of stepping into the world of research, like Taslima and myself, are very intrigued. So, can you tell us a little bit about what does the work of a molecular physiologist involve, and what exactly does your PhD work involve?
Yes, so molecular physiology is not something that I guess even biologists are not very familiar with this term, because it came up as a trend very recently, because what we are all used to is molecular biology, which everyone, even non-biologists would know about it. So it's basically study of molecules that are very key in a biological function or in a biological organism, and the functions that these molecules do, like DNA, RNA, proteins, and what do they do in the body, and what pathways do they regulate, and things like that. So that's molecular biology. But molecular physiology is one step ahead, where where the biologists look at these molecules and look deeper into the pathways and processes that these molecules regulate in a body of an organism, and how does it. So we start from the cells, go to a bit more zoomed out version, and we look at tissues, and then we look at the organism itself. So we start from one molecule, let's say a protein, and then look at the pathway that it regulates in a cell, tissue, and then how all of this affects the organism with respect to its well-being and homeostasis. So that's molecular physiology. I was also very new to this term when I was doing my PhD. I was doing my third year of PhD, that we named our lab back then as a molecular physiology lab. Until then, it was known as molecular biology, because we were just looking at pathways. But then we started looking at mice and also humans. How do they regulate and take care of these pathways? So that's molecular physiology. My work during PhD was actually a lot of things, and I'm so grateful that I did that in India, because I don't see that happening a lot in places outside, because although we here are sort of restricted with a lot of resources and everything, we are never restricted with the thought process about asking very, very intriguing questions in science. So my PhD was, so I did it at Tata Institute of Fundamental Research, Mumbai, under Dr. Or Professor now, Ullas Kultur. He was super encouraging to ask a lot of intriguing questions and never stopped us from, being innovative. So I worked on multiple different things. I was working on microRNAs, as you guys already know, and it was my chance that I hit upon that project, that microRNAs are these small non-coding RNA. So people initially call them as junk RNA, that we didn't know what they were doing. They are the small RNA which do not code for a protein. They don't have a protein function. But then later on, it was identified that they're very important in regulating a lot of processes in the cells. So we had sort of stumbled upon microRNAs as regulators of feeding and fasting cycles in mice. What we identified during my PhD was, so I was working along with a postdoc, Dr. Tantrika, who's now a professor or assistant professor in one of the institutes, good institutes in India. So we identified that these microRNAs were actually going a bit opposite to what people would think would happen during state A to state B transitions. So we look at one condition in the body of a mouse, that when the mouse is feeding a lot, you would expect some of the biological pathways to go up. So corresponding microRNAs, which can regulate these pathways, are ideally supposed to be going down. But what we found was, when the mouse was sort of transiting from a fasted state to a refeeding state, the body of the mouse, or the tissues inside the mouse, especially liver, already knew that this is going to come, that after a fasting period, there's probably a feeding coming. And the liver would take anticipatory actions with respect to upregulating some of these microRNAs, so that it can quickly sort of transit to a refeed state. So this is a bit, a lot of information for a non-biologist, but I guess to put it shorter and to sort of cut short the long story, we identified anticipatory functions that a liver can do when a body is going from one state to another. So that was kind of new, and that's the paper that I published during my PhD. So it was a lot to do with molecular biology, core molecular biology experiments, and so on. And then I was also, looking at different things during PhD, including mitochondria, the powerhouse of the cell, and various other things. But anticipatory microRNAs were my closest buddies.
So what scientific interests are you now pursuing as a part of your postdoctoral work in Denmark? Are you still with mRNA, or have you switched to something else?
Yeah, so that's something that I'm right now thinking again, because I left the field of liver, actually. That was very close to me, but I sort of, while working with liver during my PhD, I actually wanted to look more into other metabolic tissues and then understand more tissues, so that eventually when I want to start an independent research group for myself, which is what I thought I would do during my PhD, and that's exactly what I'm thinking right now as well. I wanted to understand more tissues and then link up all of these together and get more into a molecular physiology state, where I'm looking at multiple tissues and not just one, and looking into their interaction and everything to really deeply understand the biology behind diseases. So like I said, back then I was working on feed and fasting cycles, which is not anything related to clinics or, it's not a disease-oriented research that I was doing. It was just normal, basic biological function of any organism. But then during postdoc, I decided I would want to go a little more into clinical aspects of metabolic disease. So I chose a field of obesity, and then I thought I would try and understand more of fat tissue, where obesity and getting fat and overweight is becoming a huge issue nowadays, even in third world countries like India, where we are so much introduced to junk food and haphazard eating habits and so on. So obesity is what I'm researching right now, and to be more specific, I'm actually looking at how stem cells within fat tissue, which we call as adipose tissue, how do they regulate themselves to make more adipose sites, or the fat cells, which store the fat during high energy demand. So what I'm looking at is not the clinical aspect of it. Like, I'm not looking at how adipose sites are getting sick. What I'm looking at is as to the positive effects of adipose sites, where we try to understand how differentiation of stem cells happen, where they transit from a stem cell state to adipocytes, and what are the factors that affect them, molecular biology behind it, and eventually look at how these processes can affect the whole organism with respect to weight loss and weight regain and things like that. I'm primarily working with mice because my expertise was in mice earlier during PhD, but we are now trying to transit to human samples as well, because in Denmark we actually have a lot of ethical concerns, but it's also easier than in India to get human biopsies and so on. So we are doing a lot of research in human biopsies to understand these processes.